Hey guys, hope you're doing well. My name is Sanojha Rajhbavan. I'm a third year student at Charles University Second Faculty of Medicine, and I'm an alumni officer for UIMS. Today we'll be covering the topic of immunology. Immunology is a large and very detailed subject, so I'll only be covering the basics of this topic within this lecture. These include the concept of innate immunity, the complement system, and adaptive immunity. Listed below here is an itinerary of topics I shall explain in more detail. Now we will start with our introduction. This diagram shows the overall differences between innate and adaptive immunity. These include the cells involved, the time after infection, and the response time. In innate immunity, we have epithelial barriers, mast cells, phagocytes, dendritic cells, natural killer cells, and complement system proteins involved in the response. In adaptive immunity, we have B lymphocytes, plasma cells producing antibodies, T lymphocytes, and effector T cells involved in the response. Innate immunity has a much shorter response time in comparison to adaptive immunity, and adaptive immunity starts approximately 12 hours after the infection. However, innate immunity starts from the time of infection up to 12 hours of the infection. Now we will talk about the innate immune system. Innate immune responses are non-specific, create an immediate response, and have no memory cells involved. First of all, we will talk about the non-immunological defenses. The earliest protection against microbial infections involves the non-immunological defenses. Non-immunological defenses are either mechanical, chemical, or microbial. Mechanical barriers include epithelial surfaces, longitudinal flow of air or fluid, ciliary movement, and the action of coughing and sneezing. Chemical barriers include fatty acids such as lactic acid and enzymes, lysozymes and saliva and tears, pepsin in the gut, acidic pH in the vagina, and defensins being released from the epithelium. Microbial barriers include how normal flora outcompetes pathogens, producing antimicrobial substances, and how lactobacillus creates an acidic pH. Now we will talk about the pattern recognition receptors found on the epithelial cells. Epithelial and phagocytic cells recognize infections via pattern recognition receptors, or PRRs. Pattern recognition receptors detect various pattern-associated molecular patterns, or PAMPs. The innate immune system recognizes damage-associated molecular patterns, or DAMPs, which are released when damaged or necrotic host cells. The most important group of pattern recognition receptors are toll-like receptors, or TLRs. We have listed all the TLRs on the screen. TLR1 and 2 recognize several bacterial lipopeptides. TLR3 is specific for double-stranded RNAs. TLR4 is specific for bacterial lipopolysaccharides, or endotoxin. TLR5 is specific for bacterial flagellar protein flagellin. TLR7 and 8 are specific for viral single-stranded RNA. Pattern-associated molecular patterns activate toll-like receptors, which thus leads to the activation of transcription factor NF-kappa B. This causes intestinal epithelial cells to release defensins. North-like receptors, or NLRs, are cytosolic receptors that detect damage-associated molecular patterns and pattern-associated molecular patterns. NOD1 and 2 are specific for bacterial peptidoglycans and then lead to the activation of transcription factor NF-kappa B. Pattern recognition receptor MBL, or mannose binding lectin, is specific for sugar molecules containing mannose on bacterial, fungal, and viral surfaces. In the tissues, resident cells detect microbes, leading to the processes of inflammation and phagocytosis. We will talk about each of these processes in more detail. First of all, starting with inflammation. There are 5 steps involved in inflammation. Number 1 is initiation. Macrophages phagocytose the microbe and produce cytokines tumor necrosis factor, interleukin-1 and interleukin-6. Mast cells release histamine, which attracts leukocytes. Number 2 is leukocytes rolling. These cytokines allow endothelial cells to express the adhesion molecule E-selectin. Neutrophils attach to the endothelium, but blood flow disrupts this binding. Bonds form downstream, so the repetitive process leads to leukocytes rolling along the endothelial surface. Number 3 is firm adhesion. Leukocytes express another set of adhesion markers called integrins, which bind them onto the endothelium. Number 4 is leukocyte migration or movement. Leukocytes migrate between endothelial cells to the infection site by the process of diapedesis. Number 5 is action at the infection site. Leaky capillaries allow complement proteins and antibodies to enter the tissue site of infection. Here we've illustrated the process of inflammation also through diagrams. Now we'll talk about the process of phagocytosis in more detail. Phagocytosis is a process where a cell engulfs a solid particle to form an internal phagosome compartment. Phagocytes are classed as professional, and these include macrophages, neutrophils, and dendritic cells. Non-professional phagocytes include epithelial and endothelial cells. Here on the screen, we've shown the professional macrophages, how they would look like under the microscope. Phagocytes are activated when pattern recognition receptors recognize pattern-associated molecular patterns or damage-associated molecular patterns. A phagolysosome forms when a phagosome fuses with a lysosome. Degradation can occur with or without the presence of oxygen. When there isn't oxygen present, the process of phagocytosis is much slower in comparison. When there isn't oxygen, phagocytes activate transcription factors NF-kappa B and causes release of antibacterial peptides. Here on the screen, we've illustrated the process of phagocytosis. This involves chemotaxis and the adherence of microbes to the phagocyte, ingestion of the microbe by the phagocyte, formation of the phagosome, formation of the phagolysosome when the phagosome fuses with the lysosome, digestion of the ingested microbe enzymes, formation of the residual body containing indigestible material, and finally discharge of waste products. When there is oxygen, respiratory burst occurs and creates oxygen radicals that rapidly destroy foreign material found at the infection site. The granulocyte takes up oxygen and fuses with the lysosome, forming the phagolysosome. NADPH oxidase forms a superoxide oxygen radical. Superoxide then forms hydrogen peroxide. Hydrogen peroxide is acted on by myeloperoxidase to form bleach, killing the pathogen. Macrophages contain enzymes in lysosomes, leading to cell regeneration. On the other hand, neutrophils' enzymes are contained in granules, so the cell can't regenerate once these enzymes are used up. Only neutrophils produce nets, or neutrophil extracellular traps. These are useful for large and numerous pathogens. When activated, neutrophils break down their own cell membrane and extrude chromatin. The chromatin unravels to produce a trap to physically contain the pathogens. Here on the screen, we've illustrated the process of respiratory burst. The enzymes involved as well are listed on the screen. Innate immune defenses against intracellular viruses are mediated by natural killer or NK cells and interferons. Natural killer cells are specialized for killing virally infected cells and tumor cells. Natural killer cells release cytoplasmic granzymes and perforins into the extracellular space. Perforins punch holes into the target cell's lipid bilayer. Granzymes are serine proteases which activate caspases into the target cell, making it undergo apoptosis. Natural killer cells follow kill or don't kill instructions when deciding on a target cell. Natural killer cells have activation, or CAR, kill activation receptors. They recognize when cells lose MHC class I activity. This happens during a viral infection or during tumor growth. Also, natural killer cells have inhibitory or kill inhibitor receptors that recognize MHC class I activity and instruct natural killer cells not to kill the target cell. As well as these receptors, natural killer cells can also kill by ADCC, or antibody-dependent cell-mediated cytotoxicity. An infected cell with viral proteins is bound by IgG antibodies that are specific to the antigen. Natural killer cells have cell surface molecule CD16 that combine to the constant part of the antibody. This strong activating signal leads to target cell destruction. Natural killer cells produce cytokines such as interferon gamma, which activates macrophages. Macrophages enhance the action of natural killer cells. Macrophages release interleukin-15 and interleukin-12. This leads to natural killer cell activation, which stimulates further interferon gamma release. Now we will move on to the second part of the lecture. The complement system is part of the innate immune response. There are three pathways involved in the complement cascade. These are the classical pathway, the alternative pathway, and the lectin pathway. Complement system proteins are produced in the liver. Proteins circulate in an inactive form, but when activated by a foreign microorganism, they are activated by cleavage and work as part of a cascade. This is the first pathway we will talk about. The classical pathway is part of adaptive immunity. The classical pathway was identified first, hence the name. The classical pathway is made of C1 to C9. C1 binds to the FC part of the antibody once it's bound to the antigen. A conformational change causes the serine protease part of C1 to be exposed. Now activated C1 cleaves C4 into C4a and C4b. C1 also cleaves C2 into C2a and C2b. C3 convertase forms when C4b and C2b join at the pathogen surface. C3 convertase cleaves C3 into C3a and C3b. C5 convertase forms when C3b binds to C3 convertase and forms C4b2b3b. C5 convertase cleaves C5 into C5a and C5b. C5b along with C6, C7, C8 penetrate the pathogen cell membrane through the channel created by C9. This structure is called MAC, or membrane attack complex, which destroys most gram-negative bacteria. Here on the screen, we've illustrated the classical pathway's main steps. Second pathway is the alternative pathway. The alternative pathway is part of innate immunity. The alternative pathway forms C3 convertase if the classical pathway is impaired. This is a much slower process in comparison. C3b binds onto the microbe and the BB fragment binds to form the C3BB complex. This acts instead as the C3 convertase enzyme. Then the rest of the pathway follows the same as the classical and lectin binding pathways. Here we've illustrated this first different step on the screen. Finally, we have the lectin pathway. The lectin pathway is part of innate immunity. The lectin pathway has mannose binding lectin, or MBL, bind to mannose in the pathogen cell wall. C4 is cleaved into C4a and C4b, while C2 is cleaved into C2a and C2b. C3 convertase forms when C4b and C2b join. Then the pathway acts the same as the classical pathway. Here we've illustrated this first step on the screen. C3a enhances inflammation by releasing histamine. Histamine release causes blood vessels to vasodilate and become larger in volume and increases permeability to other white blood cells. C3b causes opsonization and cell lysis. Now we move on to the final section of this lecture. Adaptive immunity is made up of humoral immunity and cell-mediated immunity. Adaptive immunity has a slower response compared to innate immunity. B cells are responsible for humoral immunity, while T cells are responsible for cellular immunity. Here, the difference in cell types is illustrated on the screen. Now we will talk about lymphocyte activation. Lymphocytes are activated by antigens and undergo clonal expansion. Clonal expansion is when lymphocytes proliferate with the same antigen specificity. The immune system doesn't normally react against the host's own potentially antigenic substances. This unresponsiveness to self is called immunological tolerance. Healthy adults have 0.5 to 1 times 10 to the power 12 lymphocytes within the blood. B cells develop in the bone marrow and T cells develop in the thymus. These cells are released into circulation and then to peripheral lymphoid organs. Peripheral lymphoid organs include the spleen and the lymph nodes. Within peripheral organs, cells try to find a foreign antigen. Antigens are displayed on antigen-presenting cells surfaces bound to the MHC class 1 or 2 molecules. These MHC molecules are presented to T cells. B cells uptake antigens without being expressed on other antigen-presenting cells. then are presented to T cells. Here we can see an image of a lymph node. B cells, once activated, form a germinal center, and then B cells form the primary follicles. B cells migrate to the cortical regions, and B and T cells enter the paracortical regions. Now we talk about the initial infection and its stages. B and T cell activation takes time from days to weeks. While the body waits for the adaptive response to start, the innate immune response is vital. After fighting infection, memory B and T cells are present. So this means if the infection occurs once again, the response time is much quicker as the memory B and T cells are present and antibodies can quickly be produced from the memory B cells. The process of an initial infection is made up of 4 stages. Number 1 is initiation. Initiation is when an antigen-presenting cell displays the antigen on its surface. Antigen-presenting cells are dendritic cells or macrophages. Antigen-presenting cells can only present protein antigens. On the other hand, B cells can present protein antigens, carbohydrates, or lipids from the pathogen. Number 2 is clonal expansion. Number 3 is differentiation. This is a process when B cells produce plasma cells, and this is when T cells form either helper T lymphocytes, cytotoxic T lymphocytes, or regulatory T lymphocytes, depending on the need of the body. Number 4 is apoptosis. After a microbe is eliminated, most effector lymphocytes are induced by an infectious pathogen, will die by apoptosis. Here we see a graph showing how the concentration of antibody varies over time. When there is an initial exposure, the concentration of antibody slowly increases, then decreases, and this is the primary immune response. After a secondary exposure, the time needed for the concentration of antibody to increase to a much higher level is much quicker, and then the secondary immune response occurs. Now we will explain humoral immunity in more detail. Humoral immunity is a key defense mechanism against microbes with capsules rich in polysaccharides and lipids. T cells can't respond to non-protein antigens, on the other hand. Secreted antibodies enter circulation, mucosal fluids, neutralize and eliminate microbes and microbial toxins present outside host cells. These areas include the blood, extracellular fluid, and lumens of mucosal organs. Antibodies can't access microbes that live and divide within infected cells. Without T cell help, protein antigens produce no or a weak antibody response and are called T-dependent. Polysaccharides, lipids, and non-protein antigens stimulate antibody production without the involvement of T helper cells and are called T-independent. T-independent responses tend to show more isotype switching and usually result in IgM and short-lived plasma cells. This is illustrated on the screen on the top left diagram. The most specialized and effective antibody responses are generated under the influence of T helper cells. These include the production of immunoglobulin G, immunoglobulin E, immunoglobulin A, and immunoglobulin D. B cells act as a type of antigen-presenting cell and present protein antigens to CD4 positive T cells. If not activated by an antigen, naive lymphocytes die by apoptosis and are replaced by new cells. Naive B lymphocytes express IgM and IgD that acts as a receptor for antigens. Naive B cells are activated by antigens binding to membrane immunoglobulins. Membrane receptors have short cytoplasmic tails, so although they don't recognize antigens, they don't themselves transduce signals. The B cell receptor forms when two protein immunoglobulin alpha and immunoglobulin beta non-covalently associate. When the antigen binds, the tyrosines of immunoglobulin alpha and immunoglobulin beta are phosphorylated, which phosphorylates tyrosine residues on adaptive proteins. Phosphorylated proteins activate transcription factors involved in B cell proliferation. This is shown in the diagram in the top right. Activated B cells migrate to the lymphoid follicles. They then act as antigen-presenting cells using MHC class II for a CD4 positive T helper cell. The T helper cell expresses CD40 ligand on its surface, which binds to the CD40 receptor on the B cell. The T cell then produces interleukin 21. This undergoes the B cell to induce class switching. Now we will talk about the antibody's general structure. B cells undergo the process of VDJ rearrangement to generate a set of diverse B cell receptors. The B cell receptor is an antibody, except it has a transmembrane part that attaches the receptor to the B cell surface. The B cell receptor has 2 heavy chains and 2 light chains. The region of the B cell receptor that binds the antigen is called the Fab or fragment antigen binding region. The fragment antigen binding region is where a heavy and light chain meets. There are 2 fragment antigen binding regions on each B cell receptor. The rest of the heavy chain makes up the constant region. The 2 heavy chains are linked by a disulfide bond, and each heavy chain is linked to a light chain by a disulfide bond. Here we can see the general structure of the antibody on the screen, which includes the variable region, constant region, made up of heavy chains, light chains, antigen binding sites linked by disulfide bridges. Now we will talk about the antibody classes. There are 5 types of heavy chains which encode the classes of immunoglobulins: immunoglobulin G, immunoglobulin D, immunoglobulin G, immunoglobulin A, and immunoglobulin E. Immunoglobulin M is the first antibody response made in all immune responses. It makes up roughly 4% of immunoglobulin found in serum. When acting as a B cell receptor, it's a monomer, hence has a valence of 2. When secreted as a plasma cell, immunoglobulin M joins with 4 other identical immunoglobulin N antibodies, forming a pentamer with a valence of 10. Immunoglobulin M is the most effective antibody at activating the complement pathway. Immunoglobulin M is highlighted on the top left on the image on the left. Immunoglobulin G makes up 75% of immunoglobulin found in serum. Immunoglobulin G is a monomer with a valence of 2. There are 4 subclasses of immunoglobulin G: IgG1, IgG2, IgG3, and IgG4. The main role of immunoglobulin G is to act as an opsonin. Immunoglobulin G is good at activating the classical complement pathway too. Immunoglobulin G binds to specific antigens and allows natural killer cells to perform ADCC, or antibody-dependent cell-mediated cytotoxicity. Immunoglobulin G is the only antibody able to cross the placenta, so newborns are born with lots of maternal immunoglobulin G. Immunoglobulin G is illustrated on the bottom left on the left image. Immunoglobulin A makes up 20% of serum immunoglobulin. Immunoglobulin A is a monomer with a valence of 2. Immunoglobulin A is the main antibody found in mucosal sites, such as the saliva, tears, breast milk, and semen. Immunoglobulin A can also act as an opsonin. In mucosal sites, plasma cells underneath the basement membrane of epithelial cells produces an immunoglobulin dimer held together with a J chain. So immunoglobulin A has a valence of 4. There are 2 types of IgA, IgA1 and IgA2. Babies receive immunoglobulin A via breast milk from their mother. Immunoglobulin A is shown in the middle of the bottom row on the image on the left. Immunoglobulin E is associated with allergic and anti-parasitic responses. Immunoglobulin E is a monomer with a valence of 2, is shown on the bottom right on the left image. Immunoglobulin D is a monomer with a valence of 2. Its main role is to be found alongside immunoglobulin M, and it acts as a signal to the body that mature B lymphocytes are ready to leave the bone marrow. On the right image, we can see the structures of the immunoglobulins, and we can see how the different subclasses of immunoglobulins form a different shape and join in different ways. Finally, we will talk about cell-mediated immunity. CD4 positive lymphocytes activate phagocytes to destroy microbes that have been ingested by phagocytes or activate B cells. CD8 positive lymphocytes kill any host cell type that harbor infectious microbes in the cytoplasm. T lymphocytes only recognize peptide fragments of protein antigens that are bound to MHC on the surface of antigen-presenting cells. The process of priming a cytotoxic T cell requires two signals. The antigen is presented on an MHC class 1 molecule on the surface of a nucleated infected cell. Typically, antigens come from intracellular pathogens, such as tumor cells or virally infected cells. The first signal is the antigen binding perfectly to the T cell receptor. The second signal is co-stimulation, where a T cell surface ligand CD28 binds to the B7 ligand on the antigen-presenting cell. This is illustrated on the bottom right image. As well, CD8 positive cells can be stimulated by cross-presentation. Cross-presentation is when the infected nucleated cell is detected by antigen-presenting cells displayed on MHC class 1, later coming into contact with the CD8 positive T cell. The immune synapse is the T cell receptor bound to the MHC antigen and CD28 bound to B7. Once the signals are received, the naive T cell matures. Cytotoxic T lymphocytes release granules containing granzymes and perforins. We will now talk about the first subset of CD4 positive T cells. CD4 positive T cells are activated when bound to MHC class 2, and CD4 positive T cells differentiate into three different subsets of effector cells we should mention: T helper 1 cells, T helper 2 cells, and T helper 17 cells. First of all, we have T helper 1 cells. They are induced by microbes that are ingested and activate phagocytes. T helper 1 cells produce interferon gamma, which acts on macrophages through a CD40 and interferon gamma interaction. Binding of CD40 ligand to CD40 macrophages leads to the activation of transcription factors. Transcription factors encode lysosomal enzymes that stimulate reactive oxygen species and nitric oxide synthesis. Macrophages secrete interleukin-1 and tumor necrosis factor, which leads to inflammation and leukocyte recruitment. This leads to increased MHC and co-stimulation activity from the cell. Macrophages that respond to T helper 1 cells are called M1 or classically activated macrophages. T helper 1 cells' role is mainly against microbes such as Mycobacterium tuberculosis, Salmonella, Brucella, or Listeria. The role of T helper 1 cells is illustrated on the bottom left image. T helper 17 cells develop in bacterial and fungal infections and induce inflammatory reactions against these infections. The main role of T helper 17 cells causes inflammation by stimulating macrophage and neutrophil recruitment. T helper 17 cells secrete interleukin-7, which we see on the screen on the top right, and this stimulates chemokine production from other cells. Finally, we have T helper 2 cells, which we illustrated on the top left. T helper 2 cells are induced by parasitic worm infections and produce immunoglobulin E, mast cells, and eosinophil-mediated destruction of parasites. Interleukin-4 coats helminths and stimulates immunoglobulin E antibodies from B cells. Interleukin-5 activates eosinophils by using T helper 2 cells, FC receptors, to bind to immunoglobulin E. Activated eosinophils release toxic granule contents. Interleukin-13 stimulates mucus secretion and intestinal peristalsis. Immunoglobulin E is responsible for mast cell activation. Interleukin-4 and 13 inhibit the M1 classical macrophages whilst activating the M2 alternative macrophages. Macrophages which respond to T helper 2 cells are called M2 or alternatively activated macrophages. Here listed is a list of references from which each image from this presentation has been sourced. Now, this brings you to the end of the lecture. Thank you for your attention. This lecture has hopefully given you a solid overview to the main concepts necessary for immunology. Thank you for listening to this rapid review lecture on immunology. If you have any questions, feel free to leave a comment below.